minutes you have left, including your question time. All right.
Well, thank you, Lucy, and also Jim for organizing this important session. I think today I'm going to talk about a project in the lab, but I want to also start with thanking also my postdoc mentor, Karen Sholtoff. She's not here today. Most of the work I'm going to talk about is a project that we started together when I joined her lab, and then I'm continuing over in my lab right now, and which is basically to understand how viruses basically infect grasses, okay? And Anna mentioned of, how it's important to understand the fundamental interactions that go through, viruses, how they cause disease in plants. And in this case, I'm going to focus on that aspect and how that also, is important to also translate the findings, is important. The discoveries are important, and then there comes the translation of the findings. So without any ado, so in the audience, we don't need to emphasize on the importance of grasses. They are important because they are sources for our food and also fuel. And so the pathogens that infect grasses, including viruses, are also very significant. For example, wheat streak mosaic virus, in an example, causes severe losses in just wheat alone. That's still an old number. We also heard of some good talks on maize and maize viruses. I was happy to see a good number of talks. And that maize viruses are also important. And so also the viruses of other grasses. However, what we think is there is a bottleneck in our understanding of grass virus interactions at the fundamental level in terms of what are the defense responses, immune signaling pathways that are perturbed by the viruses in grasses when compared to a dicot virus interactions. You can see that, that there is a lot of knowledge in the dicot side, which we could not generalize all the time to a monocot. So our basically work is to essentially bridge this knowledge gap. And in our lab, we are focusing on mainly PMV, which stands for Panicum mosaic virus. It is a positive sense single-stranded RNA virus. It encodes only a few proteins, 6 proteins, and basically it forms a icosahedral virions, and you can see that 30 nanometers in diameter. And one of the interesting features about PMV is it supports the replication of a satellite virus. In this case, it's satellite Panicum mosaic virus. And it encodes only one open reading frame, and that's its code protein. And it forms about 15 nanometer icosahedral virions. So both of them together infect a wide variety of plant host grasses. And I'm just pointing out a few of them. Some of them are turf grasses, like Sintegus green grass, which is very commonly grown in Texas and also Mexico and Oklahoma, I believe. And also it infects field grasses like switchgrass. They're important for putative bioenergy crops. And also it infects proso millets and other millets as well. So millets have been, for us in the lab, the main host plant to study for over almost 15 years during the time when Karen was studying this. They were using this as a host to essentially understand the determinants of the virulence, PMV and its proteins, and SPMV, and how the synergism is working in the plants. So what is still not known is the part of the plant immune system and what is happening to that during these interactions. And unfortunately, the millets and other grasses, the grasses that I just showed you, are very hard to study in terms of the genetics. They are very complex genomes and also larger plants and difficult to grow when compared to, a model plant like Benthamiana for dicot viruses. And so we were in search for a sort of, I would say, model plant or grass for studying grass virus interactions. And in about 2010, so I think we came across Brachypodium distichiana. It was, at the time, DOE was putting forward as a model plant or model grass. For those of you who are not familiar with Brachypodium, it's basically a little grass that is in the C3 clade, very close to rice and wheat, but also close to C4 grasses. And so we thought it would be a good one. Important things about Brachypodium is it has a wide range of, community resources that are available, including a sequenced genome. And there were a collection of germplasm, but also a lot of, for functional genomic tools, there is a population of tDNA mutants that we thought would be very useful for functional analysis. And so we started basically to investigate into whether we can use brachypodium to begin with for PMV interactions. And I'm going to now walk you through some of the things what we found using brachypodium as a host for PMV and also some other grass viruses. Okay, so the first thing is we were lucky to see that brachypodium was a host for PMV, and also importantly the synergism that is imparted by the satellite virus. As you see here, basically you can see PMV causing severe stunting and also chlorosis symptoms. And with the satellite virus, the chlorosis deepens and also even severe stunting symptoms appear. And so this phenotype were almost identical to what we see in field grasses, such as this is an example from an old paper when Dr. Sheltoff was characterizing the synergism in pearl millet, and you can see the identical effects of the synergism in field grass and the model grass. So this is very important for us because it kind of gives us confidence in working with the model grass and extrapolate the findings to any other grasses like in field grasses. So the first things we did was, as most of us are already doing, is to look at, we're taking a systems approach to study at the whole genome level what are the gene expression changes that are going through during these interactions. And so for that, we leveraged the resources of brachypodium. They had a tiling array available early in 2010. We did this analysis and basically looking at global profiling, global expression changes during PMV and SPMV infection. And what was, summarizing the results, some of the things we found, what you are seeing is a overrepresented categories of genes that are induced or repressed during the infection. And there's quite a few gene function categories that are important. And without going into those pie charts, but I can just show some of those here. On your left, you're seeing a transcription factor class called proteins called WRKIs. If you are familiar with plant immune responses, WRKI are important transcription factors that mediate transcriptional reprogramming during plant immune signaling. And there are significant induction of these genes during PMV infection in brachypodium. And also we found almost 25 protein kinases that are misregulated. And many of them are also receptors on plant cell walls and solid cell membranes and could mediate the signaling to the nucleus. And we found some that were downregulated as well, but just to show a few, some of the MAP kinases are also highlighted here that are important for plant immune signaling. And so this gives us some idea of the major gene functional categories that are perturbed during a PMV interaction in brachypodium. Another way to look at the gene expression profiles is to basically correlate them to plant metabolic pathways. In this case, I'm showing you, we did this analysis using MapMan software, showing you the metabolic pathways of brachypodium that are perturbed during the interactions. And each of these pathways highlights either a primary metabolism, secondary metabolism, and the little squares in there shows genes that in those specific pathways either induced, identified by red, or green for downregulated. Just to get a quick overview, we did this and we found that several genes in primary metabolism are downregulated, including some lipid genes here, and a lot of photosynthesis and light reaction chains, reaction enzymes are downregulated. Interestingly, a significant number of secondary metabolism genes are induced. So that's one way, another way to look at some of your gene expression data. But what we're interested in is mostly defense signaling pathways, and so we focused our analysis on plant hormones and their signaling and the genes that are involved in plant defense hormone signaling. You may be familiar with salicylic acid and jasmonic acid and ethylene are the major plant defense hormones in mediating the interactions. And what we found was salicylic acid signaling pathway was strikingly upregulated, and with a huge number of genes, orthologous genes that mostly are pulled out from orthologous of Arabidopsis, or even the nearest ones we studied were in rice. And basically, most of the salicylic acid signaling pathway seems to be upregulated. And as this is a commonly known antagonism between salicylic acid and jasmonic acid. We saw quite a few of jasmonic acid and ethylene signaling genes downregulated. Just keep a note on the FAD7 in JA signaling. It's going to come back again. It is downregulated in our transcriptome data. And so to validate some of the gene expression data, we also did some metabolite profiling. This is in collaboration with Eric Schmelz's group when he was still at USDRS, and also Sean Christensen. Their group basically quantified the phytohormones and also diverse other metabolites that I'm going to show you. Well, the metabolite profiles correlate with the gene expression changes. As I'm showing you here, salicylic acid is more abundant in plants, and so is also a precursor of salicylic acid, called cinnamic acid, is also present in higher levels. And we found downregulation or lower accumulation of jasmonic acid, and also its precursor form OPDA. Now, their group also, we quantified some other hormones. In this case, I'm showing you abscisic acid. It seems to be a little bit induced. This is an important hormone for plant abiotic stress and such as drought. What we think is appears to be a crosstalk in this case. Induction of abscisic acid indicates that there is a crosstalk in abiotic stress signaling pathways with the biotic stress signaling pathways. We do have some preliminary data, which I don't have, I'm not going through now, but we found some effects of PMV also on drought tolerance, but that's for another day. Continuing on, we also quantified some fatty acids. Fatty acids are important signaling molecules in plant defense, especially insect interactions. And also they feed into the biosynthesis pathways of oxalipins, including OPDA and also jasmonic acid. What we saw was in the PMV infection of Brachypodium, we significant induction in 16-0 palmitic acid and also 18-2 carbon, linoleic acid, but there was a significant decrease in 18-3 linolenic acid. Now, remember I mentioned FAD7 as an enzyme that we saw was downregulated in our transcriptome data. That actually is an enzyme that mediates the desaturation of linoleic acid to linolenic acid. And so it makes sense to us that that was downregulated, so the substrate was accumulating, and then the product was low abundant in plants. So in addition to looking at basically phytohormones and also fatty acids, we also looked at some secondary messengers of signaling. In this case, I'm going to point out to one observation we made was in levels of hydrogen peroxide. In a typical interactions, I want to emphasize one point that this is a susceptible interaction. Okay, so usually during a susceptible or even virulent interactions, hydrogen peroxide is important for hypersensitive response or even causing necrosis. What we saw was something peculiar in PMV. We saw hydrogen peroxide levels appear to be downregulated. So here this is an assay we do by staining the leaves of infected or healthy plants with a DAB chemical. That brown precipitate indicates higher amounts of hydrogen peroxide. Basically, even if you zoom into these little the Brachypodium leaves at the cellular level, we saw that most of the hydrogen peroxide in a healthy plant was at the cell wall or cell membrane, and which is what we saw significantly downregulated. Now, what is the significance of a downregulation of hydrogen peroxide in PMV infection is a question we are still addressing. But what we have some ideas that perhaps because it is so important for hypersensitive response and necrosis phenotypes, by downregulating, PMV has a edge in the plant to be able to move forward before the plant kicks on its hypersensitive response or necrosis responses. So. So that's a little bit about some metabolic profiling work we also did. So in addition to looking at global changes in gene expression and metabolite profiles, we also started looking at really where are each of these PMV and SPMV proteins going in the plant cells, and also what are they interacting with. I don't have time, so I'm going to just skim this slide. We did localization of all the proteins, but I'm going to show you two proteins, which are the capsid proteins. And both of the capsid proteins of the PMV and SPMV appears to be localized in the nucleus. And thanks to Michael Goodin's vectors, we used his vectors to do this analysis. Basically, this is an RNA virus. Its life cycle is in the cytosol, but to us, seeing the proteins going into the nucleus indicates that there is a function of, perhaps an important function that these virus proteins are perturbing in the nucleus. And we don't know what it is, but it is a peculiar result that we saw. And so, based on some literature of other proteins that go through, other RNA virus proteins that also go through nucleus, such as a GRV open reading frame 3, which Taliensis group has shown that it goes to the nucleus and interacts with the fibrillary protein, and that interaction is important for long distance movement of GRV. We think that similar things could be going on. The PMV proteins could be interacting with some of the nuclear and nucleolar proteins too. We're still working on these two hybrids right now on that. Another graduate student in Karen's lab is working on them right now. But we have also been interested in what other things can go on in the nucleus. And as you have already seen, that there is a huge changes in transcriptome. But also nucleus is a site for post-transcriptional gene regulation like splicing and alternative splicing. And we had some preliminary data from our microarrays that we saw some splicing factors to be misregulated during a PMV infection. And so we reasoned that perhaps there is more than those, those two splicing factors could be mediating, huge changes in the alternative splicing. And in addition to that, alternative splicing is important for virus interactions. There's not that much known, but the classic N gene that you already are familiar with is also alternatively spliced. It's not much known beyond that there is two forms of N gene. So we set out to study alternative splicing changes that are occurring in plants during virus interactions. In fact, so just to give you an overview, there is different types of splicing that can occur in plants. Some of them are listed here, the major types. Exon skippings are the least predominant in plants. This is data compared to mostly dicots and also some monocots as well. And intron retentions are the most predominant alternative splicing events when compared to humans. Now, each of these different splicing types can result in changes in open reading frames encoding different proteins from the same transcript, and that can have an effect on the function of the protein itself. And so we were interested in understanding these changes during a plant microbe interaction, and so we did basically take advantage of some of the latest tools that are there. RNA-seq is useful for doing such analysis, so we went ahead and did that. And cutting down all the long story, 2 years of work and the workflow through the transcriptome and the alternative splicing analysis, what we found was essentially that there is, here I'm showing you a pie chart of the different splicing types that are misspliced during PMV and SPMV infection. So on the top here is the healthy plants. As expected, there were the majority of splicing types that are present in Brachypodium were intron retentions, and the least abundant were the exon skipping type of events. That fits sort of the patterns that we saw in dicots and also other monocots. And during an infection, we do not see a big change in overall ratios of the different types. For example, intron retentions are still the most abundant, and also exon skippings are the least abundant with both PMV, RPMV, PMV, and SPMV infections. So we think this is a qualitative effect. So there is not big changes in qualitative effects. But what is different is in each of these splicing types, there is higher number of splicing events. So that's a quantitative effect of viruses, on splicing. If you look at what are those different splicing events that are misspliced in PMV and SPMV when compared to healthy plants, and then annotate those genes based on putative functions, we see quite a few important genes in plant defenses that are misspliced. I'm showing you 3 classes. NBLRR proteins are the classic N gene, is NBLRR protein. And most of those R genes seems to be, some of them are misspliced during infection, and also transcription factors, and also splicing factors themselves sometimes get alternatively spliced. So we found a significant number of splicing factors to be misspliced. So there's a lot of data in this one slide. Each of these genes could have an impact on susceptibility of brachypodium to PMV. How is a question that is the hardest part to answer. But we started looking at one or two genes. I'm going to show you the first gene we started looking at. That's the missplicing of a splicing regulator. Now, the reason we picked the splicing factor is because splicing factors, and when they get alternatively spliced, they influence the global splicing changes. So it's like maintaining the splicing homeostasis. So basically, what is going on in this case is that this splicing factor called SCL33, which has two annotated forms transcripts in the brachypodium reference genome, a primary transcript and an alternative transcript. What we found in our RNA-seq data was a significantly more number of alternatively spliced forms, especially these intron-retained forms right here. And many of these alternatively spliced variants of this factor are also misexpressed during PMV and SPMV infection. They have a dynamic, expression or abundance patterns, that's more appropriate. So to corroborate some of this RNA-seq data, we also did some RT-PCRs really to look at the bands. RNA-seq is all numbers, but here we're looking at all the different isoforms essentially through RT-PCR, and we did identify very more than, obviously, this is our primary isoform right there, and then there is significantly more than, I think you can see at least 4 in a healthy plant, alternatively spliced forms of SCL33. And some of those are developmentally regulated. As you see, as the plant ages in a healthy plant itself, there is some abundance of these higher isoforms, longer isoforms. But note what is happening in a PMV and SPMV infection. Many of those longer isoforms, which are, I guess, developmentally regulated, are prematurely appearing. So there is a perturbation in even the developmental regulation of these transcripts in a virus infection. And we also noticed that new ones appearing in infection, like this one right here. This is absent in healthy plants and only present in PMV and SPMV infection. So we also went ahead and cloned up each one of these and then sequenced them by Sanger sequencing and looked at their essentially protein coding capacities. And we noticed that each one of these longer isoforms, because they were having some portions of introns, they end up having premature stop codons and ends up with a putatively truncated proteins. And the full-length protein of this SCL33 encodes these key domains, RRM at the N-terminus and then the SR domain. And in these truncated proteins, they pretty much lose either the full SR domain or portions of RRM domains. So what is the significance of all of this? Well, each one of these little truncated proteins could be acting as a dominant negative manner, interacting with perhaps even the targets of SCL33 or even the wild-type protein. And that can impact the function of SCL33. We don't know beyond that what are the targets of SCL33, but that's the next question we would like to answer. But just to summarize, so far what we learned with PMV and brachypodium as a sort of model for our grass virus interactions, we were able to put together some of the global changes that are going on in terms of plant defenses. As the virus, PMV in this case, enters the cell, it's recognized by plant signaling machinery through some of the, we call it virus-associated molecular patterns. And plant triggers defense signaling pathways such as SA. And as a consequence of the antagonism, we think there is a suppression of JA and hydrogen peroxide as well. And these responses are mediated to the nucleus and translates into the transcriptional changes in the defense hormone signaling genes. Now, despite this is a susceptible interaction. So we see all these defense signaling pathways triggered. They're induced, and still PMV is able to successfully cause disease. So we think there is something about susceptible interactions. They are one step ahead of plant defense pathways, either it's a premature timing of these defenses, or even somehow they are actively suppressing some of these defenses. Now, we all know about viruses and their proteins. They can do a lot of things in perturbing host cell physiology. And we think some of these viral proteins are also, in this case, PMV proteins, are trafficking to the nucleus and splicing some of these genes that could be involved in plant defense pathways. There is obviously many questions also to study further, and some of them we are interested in is looking at the splicing stories of each one of these targets that we identified, and what is their role in PMV causing PMV virulence. And also we are interested in hydrogen peroxide. Why is it? How is it suppressed, and how does that also translate into virulence of PMV? But in addition to answering those questions, we were also interested in looking at now what's going back to our, all the different grass viruses and also grasses that are important. What is going on in the other systems? And whether what we learned so far in PMV and Brachypoeum, can we extrapolate to some of the other grass virus interactions? So we basically took a comparative approach here. And we established with 6 more different grass viruses, grass-infecting viruses, barley stripe mosaic virus, brown mosaic maize mild mottle, sorghum yellow banding, wheat streak, and foxtail. We established basically Brachypoeum as well as another grass in a C4 clade, Setaria viridis, as both hosts for all these virus combinations. So the goal was to essentially compare some of the responses we saw in Brachypoeum and PMV, and compare in these different viruses and the 2 different grass types. Now, the reason we picked C4 grass was, there's not much known about how C4 specialization in plant photosynthesis and anatomies, influence the different signaling pathways. So that's one reason why we chose C4. And also the agronomic grasses that the viruses infect are C4. Anyway, so the systems are already there. If anybody is interested to study in Brachypoeum or Setaria and these virus combinations, please feel, we could talk, and we could already use. This work is published, so they're easy to infect. And we haven't tested any insect viruses though. That's another next thing. Perhaps anybody wants to study Brachypoeum and Setaria are good also models for insect viruses. Now, so basically the summary is that we started looking at one by one the different things that we saw that were changing in Brachypoeum. And the first thing we looked at was SA, JA, and ET responses. We took a qRT-PCR approach, looking at marker genes in the different signaling pathways and quantifying their expression. And I want to note that there is some conserved responses. You can see that the jasmonic salicylic acid is generally upregulated in Brachypoeum, and also just JA is downregulated in different grass virus interactions. That response appears to be conserved in Brachy and Setaria as well. Now, that's just the global view, but if you look at carefully, there is some distinct responses, like the ERF1 transcription factor right here is induced in Brachypoeum, but it's suppressed in it's repressed in here, and then it's induced here. There is a host-dependent responses as changes as well. In addition, we've noted that foxtail and wheat streak, for some reason, they clustered uniquely in their responses. So there is also virus-specific effects in the different responses we saw. So again, there is things we could tease out and study further. So we also started looking at the CL33 regulation or missplicing during the different grass virus combinations. We saw very similar results. This is a qRT-PCR, again showing the different isoforms. Many of them are prematurely accumulating those isoforms, the many viruses, just like PMV did right here. And, but some are unique only to, while PMV, for example, is able to misplace that isoform that is not present in any of those. So what are each virus is doing to, how they are missplicing uniquely is, again, another question. But, the more questions you answer, the more questions we get. So that's where we are right now to answer also some of these other questions. So wrap it up, and I'll leave. I don't have a conclusion slide, but I want to leave with this slide of an integrated plant immune model that we think viruses are important players in plant, microbe interactions. Often when reading, reviews of plant immune responses, in all due respect to the authors, viruses are excluded from the summaries, and so we sort of put together based on the existing knowledge at the time, we put together a review of plant immune responses and how viruses cause disease. There's still a lot more to update on this model, but I think in the part of the phytobiome, now being the hot topic, viruses are an important player in this microbe interactions. So with that, sorry for going through, rushing all the slides. I was excited to talk about all the work we did. I can take any questions if there is time. Oh. Totally apologize. I lost my acknowledgement slide there, but this is the most important one. Acknowledge my postdoc advisor and current collaborator, Karen Sholtoff, and also the lab, and a lot of folks who provided us with plant and virus materials and our funding agencies. Thank you.
Unfortunately, we don't have any time for questions, so we'll move
We'll
along.
talk later.
And so you can find Kranti. Thank you.